Good morning, good afternoon, and good evening to you all. My name is Anvira Sen Gupta, and on behalf of Bioprotocol Webinars team, I welcome you. Bioprotocol is an open access online journal with a mission to make research more transparent and reproducible. We sincerely thank you for tuning in from so many different places to our webinar series titled New Tools of the Trade in Life Sciences, Technologies that Enable Breakthroughs. Today, we will learn about ex vivo drug sensitivity imaging-based platform for primary acute lymphoblastic leukemia cells from our speaker, Dr. Anthony Brown, also known as Tony Brown, and our moderator, Dr. Kristen Cruz, two leading experts in the field. Before we begin the webinar, let me share a couple of quick announcements. If you have any questions related to the webinar, you can post them in the Q&A and chat sections throughout the talk, and our panelists will address them at the end. The webinar will be recorded and posted on our website later. So if you wish to revisit it, you can do so. Additionally, if you would like to receive a certificate of attendance, we encourage you to participate till the end of this live webinar. All right, now it is my distinct pleasure to introduce the moderator of today's webinar, Dr. Kristen Cruz. Dr. Cruz is a renowned expert in the field of pharmacokinetics and pharmacogenomics. Currently serves as Associate Director of the Center for Translational Pharmacology and is Clinical Pharmacogenomics Residency Director at St. George Children's Research Hospital, Memphis, Tennessee. With degrees from Rutgers University and extensive training, including residency and a fellowship in clinical pharmacokinetics, pharmacodynamics, and drug development, Dr. Cruz focuses her research on the clinical implementation of pharmacogenomics to individualized treatment regimens, particularly in pediatric oncology. The Fellow of the American College of Clinical Pharmacy and has received numerous awards for her contributions to clinical pharmacology literature. And with that, Christine, thank you so much for being here today, and I hand it over to you now.
Thank you very much, Antara, for that introduction. Thank you, everyone, for joining today. I'm Christine Cruz, and I'm joining you from Memphis, Tennessee, and I'll moderate today's session. Our speaker today is my colleague, Dr. Tony Brown. Dr. Brown is a lead researcher in the Department of Pharmacy and Pharmaceutical Sciences at St. Jude Children's Research Hospital in Memphis, Tennessee. Dr. Brown did his undergraduate studies at the Tennessee Tech University and received his PhD in Cancer and Developmental Biology from the University of Tennessee Health Science Center under the mentorship of Dr. Li-Chin Zhu. Tony now leads the work of the Pharmacotyping Resource, which performs imaging-based ex vivo drug sensitivity testing of acute lymphoblastic leukemia cells. His work has been central to the efforts of our lab to bring this testing from a research test to the clinical environment. And the topic of today's talk is ex vivo drug sensitivity imaging-based platform for primary acute lymphoblastic leukemia cells. Welcome, Tony.
Thank you, Chris. Thank you, everyone, for being here, and obviously thank you, Bioprotocol, for allowing me to present some of the exciting work going on in the pharmacotyping resource. With Chris's introduction, I don't need to reintroduce myself, but kind of just a brief overview of what I'll be going through, our recent publication in Bioprotocol about the development of this imaging-based assay for pediatric ALL, and then I'll present another paper from our group, which kind of goes how you can use the data generated from this ex vivo drug sensitivity assay to kind of help improve patient outcomes. So just to give you a brief background on pediatric acute lymphoblastic leukemia, or ALL, which you'll hear me say, it is the most common type of childhood cancer, representing roughly 25% of all new pediatric cancer diagnoses, and it estimates to be about 3,100 new cases per year. Pediatric ALL occurs relatively early on in childhood, with about 40% occurring between the ages of 1 and 4 years old. Now, for ALL, there are two main types, depending on which lymphocyte that the disease originates from, B-ALL and T-ALL, which originate from B cells and T cells, respectively. And between these two subtypes, or main types, I'll call them, there's quite a diverse range of genetic features. And I think a recent paper published by Dr. Sam Brady really highlights this diverse genomic landscape seen within ALL, with over 20 different subtypes belonging to B-ALL and over 10 subtypes belonging to T-ALL. Now, these subtypes can range just from simple chromosome numbers, such in the cases hyperdiploid or low hypodiploid. They can be specific genetic alterations, such as the cases BCR-ABL fusion protein and pH-positive ALL, or they could simply just match an expression signature similar to other subtypes, but lacking that significant fusion, such in the case of pH-like or ET1-like. So this diverse genetic landscape and biological landscape that I just talked about, this leads to a heterogeneous response when it comes to drug response, host toxicities, metabolism, which can be caused both at the somatic and germline level. This can lead to intrinsic sensitivities, drugs like LS, also causing drug resistance at relapse, can affect drug metabolism, such as the case of NUT-15 mutations with 6MP, which our lab has also worked on, or it could affect acute drug toxicities or potentially late-term effects in survivors, such as cardiotoxicity with donirubicin. And so pharmacotyping kind of investigates how interpatient variability and drug sensitivity can correlate with patient molecular profiles. And so what is pharmacotyping? Pharmacotyping is a functional precision medicine assay which tests drug sensitivity on primary patient tumor cells. And I kind of want to bring up this paper that was published by Tony Latai in 2017. With the advance in sequencing and the ease of access to genomic sequencing, it was really kind of thought that genomics and pharmacogenomics was going to be the key to unlocking precision medicine. And kind of over the more recent years, we've seen that's not really the case. Pharmacogenomics and genomic sequencing in general is really just a part of the puzzle. And here at St. Jude, we really believe that there are other functional assays that are required to kind of put these pieces together to get the whole picture. And pharmacotyping being one of these functional precision assays. So our lab here has worked on ex vivo drug sensitivity for several years. We've gone through a couple different iterations, which I'll talk about briefly. The first being this MTT-based drug sensitivity assay, where leukemia cells were plated in a 96-well plate, incubated with drug for 96 hours, and then a substrate called MTT is added, where absorbance can be read on a plate reader, which can translate to drug sensitivity. Now, this type of assay, MTT, may be similar to like cell titer glow that you may have heard of before. Robust and made into a high-throughput assay. However, some of the limitations that we saw were the large number of cells required per drug, and then it's also not really a true viability readout, with MTT measuring more mitochondrial activity and not actually telling you whether or not a cell is alive or dead. The second method that we developed was this FACS-based method, or a flow cytometry-based method. And this is where we really got into the co-culture system of adding these mesenchymal stromal cells in co-culture with our leukemia cells, where the ALL cells were plated on a bed of MSCs, incubated with drug for 96 hours, and then all of the cells were harvested, stained, and then ran on a flow cytometer. Using this method, you can use specific markers, such as CD expression markers, to identify your MSCs versus your leukemia cells. You can also use other markers such as DAPI or Nexin-5 to determine whether or not a cell is truly alive or dead. However, we also ran into limitations with this assay, and it's, again, similar to the MTT, is the large number of cells required per drug, and then due to the amount of time it took to run on a flow cytometer and data analysis, it was not really robust and capable of high throughput. So we really wanted to come up with a way to marriage these two, making a method for high throughput that is scalable, but also gave us accurate data, and we could really tell whether or not cells were sensitive or resistant to specific drugs. And so this is where the imaging-based pipeline comes in. And this is a simple flow chart on how our assay kind of works. So I'll briefly go over that. We receive bone marrow and peripheral blood samples from newly diagnosed or relapsed patients here at St. Jude. Or also from a couple of external pediatric or adult collaborating sites. The samples, both bone marrow and peripheral blood, are separated using glycol, where we try to collect this thin little PBMC layer, or peripheral blood mononucleoside layer. This is where our cells of interest are. Afterwards, RBC depletion is pretty much always done. Even if we don't have red blood cell contamination, this helps remove platelets, which is always present. Then, based off blast percent, we will either plate the sample or perform MAX, or a magnetic separation, for enrichment. And for those not familiar with hematological malignancies, blast percent is kind of how we measure tumor burden in a patient. And so we want this high blast cutoff for our samples so that we know the drug sensitivity data that we generate is representative of the tumor population and not of some other cell population within patients. After the sample is plated, it's then incubated with drug for 96 hours. A DNA staining dye and a cell viability kind of control dye is added, incubated for 15 minutes, and then these plates are read on a high content fluorescent imager, the Operetta CLS, which is made by Revity. And using these images, we can determine whether or not a cell is alive and dead, and ultimately whether or not a sample is resistant, potentially intermediate sensitive, or sensitive to particular therapies. And so I'm going to talk about the first part I'm going to talk about is the development of this assay, and I'm going to start with this MSC co-culture and kind of like why we do it. So really, there's two main functions for the MSCs. The first being it allows the leukemia cells to kind of settle to the bottom, so that way we can get an accurate 2D image of what is an inherently suspension cell line or cells. And as you can see on the image on the right, when we take a leukemia sample and plate it without MSCs, any type of perturbation caused from pipetting, whether it be adding drug, whether it be adding the DNA dye, you get this kind of dispersal of cells around where the tip went in. However, when you take the same sample, plate it on a bed of MSCs, go through the same treatment, add drug, add dye, you still see this nice even distribution of cells, which allows us to accurately get reliable data and accurate drug sensitivity data. Now, I do want to point out that this particular issue could be resolved through kind of improving certain pipetting techniques. However, the second issue cannot, and that is that the MSCs help improve sample quality over the incubation period with the drugs. We tried numerous different conditions, including collagen-coated plates, PDL-coated plates, or this is poly-D-lysine, which is a kind of a charged plate. Numerous different medias, such as MSC conditioned media at all the way up to 2%. And really, regardless of what condition we tried, we still saw this decrease in viability without MSCs. And that can kind of be seen on these graphs here to the right, where we measure nuclear area of the cells, and then also the density of the cells, and then kind of separate them based on specific gating. And our red population is red cells, and our green population is our live cells. You can clearly see that the red population is significantly higher than our live cell population. However, if we take the same sample, plate it on a bed of MSCs, you can see a significant increase in cell viability, which hopefully you can see by the increase in green objects. And one thing I want to point out, particularly about this graph in general, I want you to remember it because I'm going to mention this particular pattern later on in the talk. So now that we know that we need to use MSCs, at least for sample quality, we need to figure out the correct seeding density. We tried several different seeding density concentrations, ranging from 500 cells per well to 2,500 cells per well, all the way up to 3,500 cells per well. And these images on the top row are taken 6 hours after seeding, and then the images on the bottom are taken 4 days after seeding. And really what we were looking at is just a nice even layer, mono layer of cells, not having these holes, and also not having this overgrowth that you can see in this bottom image. Another unique thing that we noticed, especially when going into higher concentrations of MSCs, I have 3 different MSC densities here: 3,500 cells per well, 2,500 cells, 1,500 cells per well, with several different conditions. MS only, so this does not contain any leukemia cells. No drug, so it contains leukemia cells, but no drug. Ponatinib only, just with MSCs, and then ponatinib and sensitivity curves. And these are kind of mirrored group. Particular experiment was done with a ALL cell line, KOPTK1, which is sensitive to ponatinib. Basically, what we noticed at these higher concentrations is that particular drugs, and more specifically TKIs or tyrosine kinase inhibitors, such as ponatinib, dasatinib, and even sorafenib, we started to see this peeling or detachment phenotype where you can kind of see the edge of the MSC layer kind of peels up over on top of itself. And you can see it with the ponatinib only, even with no leukemia cells. However, also at the higher seeding concentrations, we did see that without drug, you still see this peeling or detached phenotype. This is due to some interaction between leukemia cells and MSCs that cause an increased proliferation of the MSCs into this peeling. However, at the 1500 cells per well, we pretty much never saw this issue. And so this is the density that we chose moving forward. Now, most of the images that you have seen so far have been using a specific DNA dye called cyclone green, which you can kind of see in figure F here, or image F. However, we chose, or we tested several different DNA dyes, as well as potential cell viability dyes, such as Cytox red, acridine orange, propydium iodide. And while in terms of accurate cell viability, this combination of acridine orange and propydium iodide, which you may see me call AOPI later in the talk, gave the most accurate data. It was not feasible to use for the entire plate. So we tested several DNA staining dyes that can really just identify the nucleus, dicycle violet, cyclone red, and cyclone green. And then we wanted to see basically if there were any differences in our drug sensitivity curves between these three dyes. And so you can see we have some drug sensitivity curves over here on the right, with cell viability being measured on your y-axis and drug concentration being measured on your x-axis. And between the three curves, you can see that there's really no significant difference between any of the dyes. So for our purposes moving forward, we went with cyclone green. This is really for two main reasons. One is the MSCs that we use for our experiment are already labeled as GFP. This allows us to keep everything within the same channel, so that in the event we want to add dyes for certain staining purposes in the future, this gives us more channels available. And then also with keeping everything within the same channel, this decreases the amount of images required for analysis, which decreases the image acquisition time and data analysis time, which is obviously helpful. So now that we know our MSC conditions and our optimal staining conditions, we needed to make sure that we could accurately separate our ALL population from our MSC population within our cold culture. So using Harmony's imaging software, or Revity's imaging software called Harmony, we can provide an input image that is taken with the operative CLS, use certain building blocks such as fine nuclei, which is able to accurately label the nuclei that the cyclone green highlights. We can measure certain properties from these objects, such as nuclear area, nuclear intensity from the dye. And then using specific gates, we can kind of separate our populations, with, again, the green population being our live cells and the red population being either dead cells or MSCs. And for this particular case, we used an area of 130 and a mean intensity of 2800, which is important for the next one. So one of the potential problems that we ran into with this manual analysis is that not the area and mean that we use for cutoffs is not going to work for every single sample. As I mentioned at the beginning of the talk, that ALL is quite genetically diverse. We can have increase in chromosome numbers, which could lead to an increase in uptake of dye, increased nuclear size, etc. And so these cutoffs aren't going to be able to apply to every sample. And so for this particular sample, we had to change the area and mean to kind of be lower for each, with smaller nuclear size and less intensity uptake. The issue, maybe not particularly for this case, because you still see a beautiful separation of your live cells, your dead cells, and your MSCs. However, if you recall one of the earlier images, I showed you that there was really just a range of leukemia cells. You didn't really see this beautiful separation of your population. And due to this, it could potentially lead to some bias depending on what cutoffs or what gates the person doing the analysis picks. I might pick 2000, another person in the lab might pick 2200, etc. And so to kind of eliminate this human bias, one of the senior researcher in our group, Landon Choi, came up with a machine learning pipeline, again, using the imaging software from Revity called Harmony. And similar to the manual, we can provide an input image And however, this time we just changed the building blocks. Instead of find nuclei, where it only identifies the nucleus of a cell, we use this find spots feature. And this pretty much allows us to not only label the nuclei, which hopefully you can see here, but also allows us to pick up other random objects, potentially just random artifacts. It could be dead or dying cells. It could be extracellular vesicles, anything that could potentially influence our results. We then use this remove border object feature because for our images, we take 9 separate fields of view and then stitch them together to form a single image. We want to make sure we're not counting any objects more than once. Once we have all of our objects, we can calculate certain properties such as intensity, area, morphology of the object, certain surface textures, really as much information as possible to provide to the model. Once we have all of that calculated, we then train the model by basically identifying whatever populations of interest that we want. And then it takes the information that it's gathering from those objects to basically learn from that. So in the future, it can identify them without us telling it. And so this machine learning model was created on a data set of 10 separate cases with 10 objects per case. It's for an N of 100. The reason we chose this is just from the historical literature. This was the recommendation for creating a machine learning model. If you choose more than this, it actually kind of creates bias and you end up with more error, even though you think it's a better trained model. The images were picked at random to help remove any bias. And then once the machine learning model was made, it was ran on 2 additional data sets to kind of verify the results. But ultimately, we ended up with 4 different populations: a live ALL1, a dead/artifact population, an MSC population, and a live ALL2 population. And the reason we have a live ALL1 and a live ALL2 population is just so that, as I mentioned, the genetic diversity, we also see that phenotypically, and this allows us to capture all potential cases that we have seen so far. So now that we created the machine learning pipeline, we wanted to kind of validate it. And so we started at the macro level. So basically just looking at LC50 curves or drug sensitivity curves to making sure that the machine learning pipeline was basically representing what the original manual pipeline was outputting. And so these are drug sensitivity curves similar to the previous with percent survival or percent viability on your y-axis and drug concentration on your x-axis. And you can see that between both models, they basically mirror each other and are almost identical. We then wanted to kind of validate this at more the micro level. So instead of looking at a whole drug curve, looking at individual wells within a drug curve. So we do 6 concentrations in duplicate. So ultimately 12 wells per drug over the data set of 10 cases. So over 120 wells per map. And so what we wanted to look at was the viability that we were picking up in an individual well. Is it the same or is it at least similar in the machine learning compared to the original pipeline? And so our machine learning is on our y-axis and our original manual pipeline is on the x-axis. And hopefully you can see from the R-squared values that they correlate beautifully across a wide variety of different drug mechanisms, cytotoxic drugs, such as daunorubicin, L-asparaginase, prednisolone. You have certain tyrosine kinase inhibitors, such as fostamatinib or dasatinib, and even BH3 mimetics, such as navitoclax. The machine learning pipeline works absolutely great. Finally, we then wanted to validate the imaging pipeline as a whole compared to our original MTT and flow-based assays. The top 2 rows are going to be in comparison to our MSC flow-based assay, which was our kind of second iteration. And then the bottom 2 rows are going to be against our MTT assay, which is the first iteration. And again, you can see with the R-squared values that they correlate quite nicely. And so this really gives us confidence in using this imaging-based assay for ex vivo drug sensitivity moving forward. And so to kind of give an overall view of what our current drug panel looks like, we currently test 40 drugs with a wide array of mechanisms, such as cytotoxic agents, even monoclonal antibodies, such as inotuzumab, targeted therapies, such as dasatinib, ponatinib, et cetera. And this is kind of what our output looks like on the right with the drug sensitivity curve. Don't worry about any of the numbers. They're randomly generated de-identified numbers. But basically what I want you to gather is that we do everything in duplicate, and we get very nice consistency across all of the drugs we test. And so that was pretty much how we went about developing this imaging-based pipeline. Now I kind of want to talk about how data generated from pharmacotyping can kind of be leveraged to improve patient care. And so this was another paper published by our group here at St. Jude that used pharmacotyping data to identify a subset of TLL cases that were sensitive to targeted therapies that previously weren't known. And so dasatinib is a drug that targets BCR-ABL positive as fusion protein that's found in pH positive BALL exclusively. And you can see with this graph on the right that cases that are positive for this BCR-ABL fusion are exceptionally sensitive to dasatinib. However, looking at our pharmacotyping cohort, especially for TLL, we noticed that a large population, especially in pediatrics, where it was over 40%, was also uniquely sensitive to dasatinib, even without this fusion, and even to the same levels as pH-positive BALL. So to kind of systematically examine some of these biological factors that could potentially be underlying this TLL response in dasatinib, we took RNA-seq data from a TALL cohort called Target TALL and created this kind of RNA-seq interactome where we identified hub genes as well as potentially interacting genes to kind of look at more like a network analysis. And this was able to identify over 27,000 hub genes and over 1 million different interactions. We then took the RNA-seq data from our dasatinib response cohort for TLL, both resistant and sensitive, and overlaid it across this network to see if we could identify any driver pathways in both the resistant cohort and the sensitive cohort. And so I'm going to start with the sensitive cohort. What we noticed was an upregulation of this pre-TCR or T-cell receptor activation pathway. And in particular, these genes LCK and PTCRA were some of the top activated genes in our sensitive cohort. We then took dasatinib and then ran it through 3 different TKI databases and saw that LCK actually can be inhibited by dasatinib. We then created a dasatinib biomarker score, which you can kind of, you might hear me say LCK activity or dasatinib sensitivity. They all are kind of used interchangeably. And so if I say one, just know that I mean them all to be the same. But looking at the dasatinib biomarker score, we put it across TLL subtypes, and what we saw was a pretty wide range across TLL subtypes, with this potential dasatinib sensitivity score being high in TAL2 and TAL1 subtypes, and then kind of decreasing as you reach HOXA-A or TLXA3 subtypes. We then noticed kind of an exquisite sensitivity in a recently described TLL subtype called SPY1. And that subtype showed that at the expression level of PTCRA was associated with T-cell development stage. And so we looked at RNA-seq data for 19 different stages of T-cell development and saw that the dasatinib sensitivity score also increased as you increase during the T-cell development stage, with it being low in this early T-cell precursor stage and then getting higher as you reach this double-negative-3, double-negative-4 stages. We then looked and see if this biomarker score was associated with survival. And what we saw is that the biomarker score was negatively associated with event-free survival and treatment outcome in this target cohort. And then in contrast to that, in the resistant network, we saw that BCL2 were some of the top activated genes within this resistant cohort. And for those unfamiliar with BCL2, it is extremely important in mitochondrial apoptosis pathways and can also be targeted by specific drugs such as called BH3 mimetics, such as venetoclax, which we had on our pharmacotyping panel. So based off this information, we hypothesized that our dasatinib resistant cohort should be more sensitive to venetoclax compared to the sensitive. And when we looked back at the pharmacotyping data, that's exactly what we saw. The overall venetoclax LC50 and the dasatinib resistant cohort was significantly lower when compared to the dasatinib sensitive cohort. And then when we looked at TLL subtypes and T-cell development stage, we also saw a remarkable negative association with BCL2 activity and LCK activity across subtypes, as well as across T cell development stage when looking at this early T precursor to the DN3, DN4 stages. And then finally, we took PDX cells from a TLL patient treated with both the vehicle as well as dasatinib. We separated out the live or viable cells, performed single cell RNA sequencing analysis, and then using unsupervised clustering, we identified 3 different clusters, C1, C2, and C3, which you can see the heterogeneous makeup of each cluster in the stack plot to the right. And basically what we saw, similar to the inter-patient variation, but at the intra-patient or single cell level, we still saw this negative association of LCK activity and BCL2 activity. And what this leads us to kind of believe is that this provides rationale for a combination of both dasatinib and BCL2 inhibitors in TLL. And so kind of to summarize this paper, pharmacotyping was able to reveal a subset of TLL cases that were sensitive to dasatinib. And I want to highlight that this sensitivity could not be identified just from genomic sequencing. We needed the pharmacotyping data in order to identify this population. And that sensitivity to dasatinib was due to LCK activity and was found to be inversely proportional to BCL2 activity. And then finally, the intra-patient heterogeneity that we see to dasatinib venetoclax response kind of leads us to believe that patients with TLL could benefit from this combination. And really to highlight to that note, venetoclax and dasatinib are now being used in a clinical trial here at St. Jude for relapse and refractory ALL. And kind of to provide an overall summary and future directions, we developed an imaging-based ex vivo drug sensitivity assay that was able to improve upon our current MTT and flow-based assays, both in scalability, the amount of sample required, sample processing time, sample imaging or data acquisition time, really all aspects. And then the drug sensitivity results that we can get from this ex vivo assay can be leveraged to identify novel sensitivity patterns in ALL subtypes. And that the combination of pharmacotyping with genomic profiling really creates a powerful model for pharmacogenomics research moving forward. And then as far as future directions go, I really want to point out 2 papers that were recently published, one from Dr. Dolman's group in Australia and the other from Dr. Azam's group in Florida. And both of these papers used a pharmacotyping assay in the context of a clinical trial to kind of guide patient therapy. And while both of these papers used a different model of ex vivo sensitivity compared to ours, I think both are equally impactful and both really show the direction that precision medicine is moving. Sciences Department, Dr. Young, Dr. Cruz, Dr. Molinelli, and then the University Hospital in Zurich, JP's group, who was absolutely instrumental in helping us set up this assay. And I'll be happy to take any questions.
Thanks, Tony. So if you do have a question, feel free to type it into the chat, and I can see the chat and I can read those questions to Tony, so you don't have to manage both screens. So Tony, there are a couple of questions coming in. So the first question is, your talk focused on studying ALL cells. So the question is, how much time and effort do you think one needs to invest to apply this pipeline to other cancer cells?
So for our particular pipeline, it's a great question. If we started out with other hematological malignancies, we are looking at focusing on or moving towards AML next. Other hematological malignancies might be easier to work with if we can get the tumor sample from the peripheral blood or bone marrow. Then it might be something that is easily applicable. For other solid tumors, it will take a little bit more work. If the papers I mentioned, let's go back. Here, they focused both on blood cancers as well as solid tumors. And this paper from Australia, they did a pretty extensive culture. It really kind of seems like for solid tumors, you need to culture the cells for a little bit before you can just immediately go into the assay. Whereas for the hematological malignancies, we can kind of just go straight from peripheral blood or bone marrow straight to the drug sensitivity assay. So it's a little bit easier. But once you develop the actual process for it, it does seem within the same genre. So blood cancer to blood cancer, solid tumor to solid tumor, it is a little bit easier to move across those.
Great. And then there's another question that came in. You focused on the imaging-based platform that we currently use, and you talked about the previous platforms that were used also to do similar assays. So a question came in about how accessible is this technology, and how could someone maybe with fewer resources launch a drug sensitivity assay?
Yeah, that's also a great question. Specifically for the imaging based, I'm very fortunate to work at St. Jude, where we have great funding and great support from the hospital. The imager itself it can be quite expensive. However, some of the other two methods, a flow cytometer, has gotten, I don't want to say cheap, but cheaper in the context of equipment as time has gone on, just with the ease of access and availability to them. The more easily accessible method would be this, plate reader. You could do this with MTT-based assays. You could do cell titer glow based assays. And while I did point out that it's not really a true viability output, cell titer glow is kind of the gold standard for doing cell viability assays. And so while we were kind of being a little more specific and stringent with our results, you could easily do this and still get accurate cell viability data using a plate reader. And so using this type of instrument and this type of assay, I think it could be easily accessible.
Great. Another question came in specifically about the culture. The question is, have you tried Matrigel for the culture?
So coincidentally, so not for this, we have not tried Matrigel, but so when I got my PhD, I did use Matrigel for a lot of 3D cell cultures with hepatoblastoma and hepatocellular carcinoma. While that is possible, it could provide good support for the leukemia cells. I'm sure whoever asked that question, Matrigel is not cheap. And so doing an entire 384 well plate, and then for our clinical assay, we run 4 plates per sample. It adds up. And so Matrigel, while realistically probably could provide that support, is probably not feasible in terms of what we're trying to do in scalability.
There's a question, Tony, about do you work with fresh or frozen cells, and is there a preference?
So I'll say technically have worked with both. The preference is definitely for fresh. If you ever try to culture a cell line from cryo, you always end up with dead cells. Hopefully the cell line recovers just fine. It's kind of a similar process with patient samples, except I would say they're honestly even trickier. When it comes to cryo samples, it's really just a 50-50 shot whether or not that sample is able to survive the conditions from being cryoed and thawed. And then even if it is able to survive, we do see a lot of variability within the data, across different wells, and as well as the potential effects on MSCs. So while it is possible, and we have, for certain cases, have gotten good data from cryo samples, I would say fresh is almost always the best way to go. And that's kind of one of the benefits, at least working with ALL for now. Obviously, doing a bone marrow is not exactly non-invasive, but sometimes if it's a high blast percent in the peripheral blood, getting a peripheral blood sample is usually relatively harmless.
Yes, and I'll just add to that answer that we do prefer, our lab does prefer to work with fresh cells. We do partner with research partners from around the country, from around the US, and we've been very successful in receiving shipped samples that were drawn fresh, shipped overnight to us, and we've successfully worked with them. I think, Tony, through about 4 days old, and we can still work very successfully and run the assay, starting to assay them at 4 days old or fresher. There's another question that came in the chat about the machine learning algorithm. So how long did it take to train the machine learning algorithm to get a reliable cutoff? Again, asking to think about applying this to other cell systems.
Yeah, that is a great question. I'm sure it's something, I don't know if Landon's in the chat, but something that he'd probably be a little better to answer. But I do know it took it took a little bit of time. I think he's much better at it now. We're currently working on, or he's, I would say he is currently working on a machine learning pipeline for, for one of our controls. He also, I kind of, made it look super easy as far as what he did. You'll notice this says 2.5 right here. He went through, I think, 4 or 5 different models, trying to get this to work and trying to get the best results, where, he would change the number of cases it was trained on, the N, or like the number of objects it was trained on, et cetera. And so, I obviously can go through and make his work look quite easy. I think starting off, especially if you've never used Harmony, if you're new to machine learning, there's definitely a learning curve that has to occur. But once you use it, the software that Revity has created is fairly intuitive. And I think, once you get your first one, it's kind of easy to build off of that.
Thank you. Those appear to be all the questions that are in the chat. Does anyone else have another question for Tony, our speaker? I'm not seeing any other questions. So I'd like to thank Tony Brown for speaking on this topic today and for bringing this to everyone's attention. Thank you very much for your seminar, Tony.
Yeah, thank you everyone for having me. Thank you everyone for attended. And thank you, BioProtocol, for allowing us to give us a platform for some of the amazing work that we're doing.
Thank you so much, Tony and Christine, for the insightful talk, and thank you everyone for coming, joining, and asking great questions. It was really a learning experience for all. As I mentioned earlier, the webinar is being recorded and will be posted on our website later. To stay updated on BioProtocol's latest journal, release articles, as well as the webinars, you can visit our website and also follow us on LinkedIn and X. Thank you everyone for coming today, and hope to see you again next time.
Thank you.
Thanks, Antara.
Now the questions are coming in.
Yeah, no, I saw that. If all these people are still on here, there's a couple I can go ahead and, I think, Ivana. So for the AML pipeline, so realistically, AML does look slightly different, like under morphology wise under a microscope. A lot of the cytoplasma to nuclear ratio is a little different, and they're ultimately larger. So really, I mean, we've been able to test a couple with cell lines and played around with, a couple of patient samples. And really all we saw is that essentially the nucleus, because the cyclone, stains specifically the nucleus, we saw an increase in size, and that was pretty much it. As far as all the other conditions using MSCs, the AIM-5 media that we use, the serum-free media, that all pretty much worked the exact same. And so I think it's going to be fairly easy for us to transition to AML as well. but that's also just kind of anecdotal. We haven't tested an extensive population of AML patients. Let's see. And then I think one person, DC, future oncologist, would guide chemotherapy based on this kind of assay. That is an excellent question, and kind of one of the things that we're working for on us trying to acquire clinical or as CAP and CLIA certification so that we can move into a clinical light. When it comes to, so I can only really speak on ALL and what we have done so far, and that might be kind of tough as far as diagnostic cases. Here at St. Jude, we've developed, may not know, but we have this protocol called total therapy, and it works quite well overall. We have, I think it's like a 94% success rate or 95% success rate for pediatric ALL. And it's going to be hard to suggest to a clinician like, hey, I know you have this regimen, but our assay says something different. You have a 95% success rate with this one, and I can't necessarily tell you what the success rate is for this other one. So in terms of diagnostic cases, it's going to be tough, and there's going to be a lot of data that's going to have to be shown. In terms of relapse and refractory cases, yes, absolutely. The two papers that I mentioned at the end, that's what they focused on, was high risk relapse refractory cases where these patients have tried every standard chemotherapy, and nothing has really worked. And so when cases like this, yes, absolutely, I think oncologists would and have used this type of assay in the future.
Okay, thank you so much. I also want to say that Nandan answered parts of the question regarding the machine learning algorithms, so we will also post that on the website later. Thank you so